Daniel Gonzalez II, and I'm an incoming environmental science major participating in the Summer Bridge to Research program and currently working under Dr. Ying. Today, I will be discussing my time researching how managing plant microbe interactions in various production systems can promote plant health and sustainable agriculture. So one of the biggest questions we currently face is how can we sustainably feed the next generations of people as world population continues to grow and the existing forms of crop production only become more dated and less efficient. With the excessive amounts of cost in both energy and money comes a great amount of agricultural waste output. So with this in mind, wouldn't it be cool if we could reuse such material to potentially improve the quality of our soil along with the amount of our crop yield? Agricultural waste materials can be converted into several biological high-value targets capable of promoting plant growth, and billions of pounds of agricultural products waste are generated annually, some of which can be utilized to produce substances like biochar or pyroligneous acid. Previous findings show that PA and or biochar mixed with soil can act as efficient forms of biopesticides and plant growth enhancers. This would be a much better alternative to conventional substances such as synthetic chemicals or fertilizers, which are much more expensive and negatively effective towards the environment. So this little picture I have right here kind of just displays the process in which we can use agricultural waste materials to develop biochar and pyroligneous acid. Basically, you put the waste material into a furnace and you burn up the material so much that it becomes a form of charcoal. But since that charcoal is coming from a biological substance, it's a biological charcoal, hence biochar. And the gases that are produced in this process can be captured as aqueous substances, and that's what's pyroligneous acid. So in addition to the benefits you see over here, biochar and PA can also promote the lengths of shoots and roots and the individual facilitation of seedling growth, according to a 2019 article. So the objective in this study is to examine the effect of applying to soils biochar and PA produced using agricultural byproducts, in this case, almond shells, on citrus plant growth, soil carbon and structure, and changes in the composition of soil microbiota through a variety of chemical analysis, including water extractable organic carbon, which can infer the amount of available carbon there is for microbes during a soil wetting event. We wish to see if regulating microbial interactions in field and indoor production systems will allow us to further promote plant health and sustainable agriculture. Because understanding the dynamic between soil nutrients and neighboring microbiota after application of biochar and PA will allow us to better assess the efficacy of these amendments to stimulate plant growth, promoting rhizobacteria and plant diseases control mechanisms. And all of this would be a significant component in evolving from a linear economy to a circular economy where much less resources, energy, and money would be wasted. Ultimately, there'd be an increase in the sustainable intensification of farming, as we see over here. So we can split up our experimental design into two major sections. The first being solid phase sampling and processing, and the second being water extractable organic carbon and microbial biomass storing. But before we can get into either of these processes, I must go over the treatment setup prior to the sampling. So all of the different soil treatments for each respective citrus variety, since we are dealing with C35 citrus and Carrizio citrus, varied in the levels of mixed concentrated PA, biochar, or both substances. And with the amendment levels of biochar being 10% by mass and the amendment levels of pyroligneous acid being 0.1% by volume. And all of the 24 different treatments were also split up into two different irrigation systems, open and closed. So here's a little graphic that I made, kind of skimming through both of the processes or major sections. So first we go to the greenhouse and we sacrifice 2 cones from each of the soil treatments and we combine them into one bag. The reason why we combine them into 2 cones into one bag is to make up for the limited amount of soil we're dealing with. Afterwards, we use forceps to remove plant material from the soil and we put that in a separate bag. Following that, we place those bags, all of those bags, into a refrigerator where they are to be refrigerated for quite some time. Once they're ready to be weighed out, we take out the bags of soil, we homogenize the soil that's in the bags, and we weigh out the soil for water extractable organic carbon and microbial biomass analysis. And the analysis we conducted for both of these substances were based off of the methods in the studies of Fier and Schimel in 2002 and Hamyak in 2018. So what we're looking at here is the mean value amounts of water extractable organic carbon and microbial biomass for all of the past months so far, but just only for the C35 citrus variety. If we are to move on to the next slide, it's just going to be all of this data plotted onto two separate bar graphs. With the x-axis being the different treatments and the y-axis is xi, I believe, being the abundance in water extractable organic carbon or microbial biomass. So if we take a look at figure 1, we see that there is a gradual decline in the amount of water extractable organic carbon across all systems over here. One possible reason for less carbon as the months pass by is that some of that carbon might be seeping out through the soil cones in each of the respective treatments over time. Another reason is that if there are more microbes over time, then the water extractable organic carbon substrate would be consumed at a much faster rate. If we move on to the second figure, we see that there is a definite change in the microbial biomass. However, we can't identify exactly why. The closed systems with both biochar and PA over here show to be the most promising conditions for the microbes. And we believe that's because biochar is capable of providing a good habitat for neighboring microbes, and the PA mixed with the weekly watering most likely provided for the best form of transport for the microbes to move around and consume the remaining water extractable carbon substrate. So to wrap this all up, according to the general trend, water extractable organic carbon is decreasing over time for either one of two reasons, because the carbon is leaking out from the bottom treatments over time, or because since there are more microbes, the water extractable organic carbon substrate would be consumed much more quickly. What was originally hypothesized was that there would be a parallel increase of beneficial, naturally occurring microbes in the nutrient, along with the decrease in bacterial diversity, which would have confirmed that it is both safe and efficient to apply these amendments, PA and biochar, to stimulate plant growth, promoting bacterial species and plant disease control mechanisms within the soil. However, we can't say there was an increase in naturally occurring beneficial microbes because we don't know what is exactly contributing to the microbes. There is definitely a change, though, occurring in the microbial biomass. And let it be known that this project is still ongoing and nowhere near being complete, since we still have a couple of months of sampling to do. But we are very excited for possible future findings. I would like to take this time to thank the Soil Biogeochemistry Lab, or also known as the Dirty Lab. I would like to thank the grant donors for this project, the USDA and NFIA. I would also like to thank the RISE peer mentors, Isai Gonzalez, Salim Zurab, John Sedrak, and Veronica Piñata, and also the RISE coordinators, RISE funding donors, and just RISE in general, along with the College of Natural and Agricultural Sciences and the Summer Bridge Research Program. I'm very grateful to be here, and it's honestly been one of the most enlightening experiences I've had as a scientist. So I'm very grateful for that. These are my references, and this is our contact information. Thank you.